Okay, our next speaker in the symposium is Catarina Lira Medeiros at the Botanical Garden of Rio de Janeiro, and she will be talking to us about the epigenome of tomato and its implications for the domestication process of this species.
Hi, good morning. Thank you for coming on our epigenetic session. So this is a work that I did in my postdoc while I was at the New York Botanical Garden with Emily, and now she's at the University of California, Riverside. And I stayed there for 1 year, and we did the bisulfite sequencing of tomato. So I will talk more about it. So our project was focused on plant adaptation and trying to explain and study the plant adaptations and the evolution of the plants in a broader way, regarding the epigenetic variation of the plants. So I brought here pictures of really classic plant adaptations, like desert plants, cactus, really adapted to dry areas, a lot of sun. Carnivorous plants. So at the Botanical Garden in Rio, we have a special greenhouse for carnivorous plants. There are many beautiful different plants, species. And of course, I bring up the mangrove plants that I've been working with mangroves for a few years. So I have these two special publications about the epigenetic diversity and the genetic diversity of one mangrove species that has a really striking different morphology depending on the localization inside the mangrove area. So this is the mangrove area I've been working. I've been doing this work just to show you about the variety of locations and environments. So here is a salt marsh plain and the mangrove plants here growing nicely. We have also the river area, river flow and the mangroves around. And we have this really sandy with areas with sand because of the river and the mangroves around. And we have really areas where it gets really dry when it does not have the tides coming and the mangroves there. So those are beautiful plants to study adaptation. So this one doesn't grow too much, but it has flowers in it. I don't know if you can see that. So this one has the pneumatophores for adaptation to grow in this kind of soil. This one has other kind of roots, the aerial roots, and also, of course, the salt excretion and secretion. So they have different mechanisms for similar adaptations, basically. So back to the subject. So I have been working with mangroves a lot more time than tomatoes. So it's really interesting. I decided to work with tomatoes just to have the other side of the plant adaptation that is based on domestication. So here are a few examples of wild species, probable progenitor species of the domesticated species. So you have here the corn, the potatoes, bananas, and tomatoes. So usually during domestication, phenotypes that are more interesting for agronomic, so for the agronomy, but also for the cultivation of this crop species and for the consumption of these species, they were selected, artificially kind of selected during time, and now we have these domesticated species that are really the fruit especially looks really different from the progenitor plant, the wild species, and also other traits, not only the fruits. So domestication is a really important evolutionary process. Darwin dedicated a chapter to variation under domestication, where the origin of the species, where he stated that it's really important to study the variation of the domesticated plants to better understand the plant evolution. So nowadays we have a lot of really exciting new techniques to study genomics and SNPs, EpiGBS, so really exciting growing field with new techniques for both genetic variation and epigenetic variation study on plants. And crop plants, they domesticated plants, they have a lot of scientists study these plants, and they are really, a lot of them have already been sequenced, or it has a lot of data available, transcriptomes. So it is really nice to work with domesticated plants because of all the tools that you can use to analyze your data. So those are two really good recent reviews on domestication and variation in plants. So epigenetic variation, genetic variation, epigenetic variation, they have sort of, now that you've seen the couple of presentations before me, I can say that epigenetic variation is usually, can be triggered by environment change and can change molecular and cellular process. So we believe that both genetic and epigenetic variation can create new phenotypes. Epigenetic variation, we say that it's potentially inherited, but we have a few examples that part of the epigenetic variation is inherited. And so this is enough for the adaptation and evolution of the species. So I'm focusing on DNA methylation. It's really important for genome stability and regulation of gene expression. And I assume, so the DNA methylation variation that we find is just a small part, or a part, not small, but a part of the whole epigenetic variation. Like Joshua said, we have interference RNAs, we have other types of epigenetic phenomena on the organism. But basically, cytosine methylation can change or control gene expression and transposomal elements. And also, we know that a part of the epigenetic variation can be inherited. So here you can see that the environment, usually, it can influence the epigenetic variation. Epigenetic and genetic variation, they have a feedback. And a part of the epigenetic variation can be inherited. And so it can have a big role on plant evolution and adaptation. So my hypothesis is based on all this knowledge on epigenetic variation. So we believe that DNA methylation states, like if it's methylated or not, so cytosine methylations were fixed after the domestication in Solanum lycopersicum, the domesticated tomato. So we decided to work with domesticated species of tomato, Solanum lycopersicum, and the wild relative possible progenitor, Solanum pimpinellifolium. So here you can see the huge difference on tomato size, for example. So I use bisulfite sequencing. So I treated DNAs with sodium bisulfite, and so the methylated cytosines, it stays as cytosines. And the cytosines that are unmethylated is changed to uracils and then to thymines. So after we do the construction of the libraries and the sequence, and when we have the data, this program, Bismarck, is really easy to use. It has online plants. So since tomato has already been sequenced, I use the reference genome of the Solanum lycopersicum species to align my sequencing using Bismarck. So Bismarck is already made for bisulfite treated data, DNA. So it already shows you areas that were cytosines that were methylated, and it separates by contact, CpG, CAG, CHA. So it's really easy tool to use, really good. And then I analyze the data. So I used 2 accessions of domesticated tomato, lycopersicum, and M82 and Elsa Craig. And I use here is the M82 fruit and Elsa Craig fruit. They are a little bit different. And I use 3 accessions of the wild Solanum pimpinellifolium. They were all seeds from the germplasm bank at UC Davis. And those 3 accessions from Peru, I believe that they are like, they could be interpreted as wild populations of the wild tomato. And we decided to use 3 accessions because we thought that they might have more variation because they are wild species. So we designed this is our experimental design. We used 2 biological replicates each accession, so 10 plants total grown in the same conditions. And the tissue we use for the libraries, for the sequencing, was fruiton breaker stage. And so we used high-seq. We had 46.5 gigabase after the cleaning of the sequence, so just the good sequences here. And after Bismarck, we have a mapping efficiency that vary between 42 and 60%. But the reference sequence of tomatoes still has a lot of ends on the chromosomes. So I haven't done any statistical analysis about that, but I'm pretty sure that I have covered much more than only 42 or 60% because we have huge amounts of areas with not well annotated on the reference genome. So they created like 1 chromosome called 00 that are the sequence that is not on the it's not yet placed on the 12 chromosomes. So this is the total of cytosines that we got on our sequencing. We had a lot more data from 2 domesticated accessions than the 3 wild accessions. So 60 million cytosines and 80 million cytosines on domesticated, and around 10 million or 9 for the wild species. It's a lot of data. So per context, we see that most of our cytosines are not on CpG or CHG context. They are just cytosines, CHA, just cytosines all around the genome. So most of these divergence on the cytosine number comes from probably, it could be a difference because I use the reference of genome of the domesticated against the wild, although they are really similar, and I'm not sure if it's really a difference of the whole genome composition, or if it's a method, something because of the method. I need to check that with more carefully, I'd say. But CPGs and CHGs are the most important cytosines to analyze for the epigenetic markers. So we got around 2 millions of, more than 2 millions for the domesticated, and more than 1 million for the wild to analyze for the methylation. So the total methylation we found, so this is really the total, it's not the percentage, but we found, if you see back, we have a total of 2 million, 2.6 million here, for example, for Elsa Gray. And of these, 1.9 and 2.4 were methylated. So we got a lot of methylation going on these two contacts, CPG and CHG. And the CH8, curiously, I had no methylation at all, all genome. This is after I cleaned the data, so I kind of get rid of all variation. So they have methylation, but they have so much variation that probably my duplicates are differentially methylated, so I excluded them. I need to analyze in a different way to get the amount of this methylation here. So that's it, all were unmethylated. So this is really curious because, so we had the fraction of the methyl cytosines per contact. We have half, like M82 was almost half CPG and CHG. I'm sorry, this is so difficult to read. But blue you have CPG, and red you have CHG, and CH8 is 0. So for most of the species we have more CHG than CPG, but everything around 50 something, 40 something. But it's really contrasting to what have been published for Arabidopsis and for tomato very recently in 2013. So they found for Arabidopsis most of the methylation on CG sites, and the rest, like half on CH8 and half on CHG. And for tomato, it's even more different from my results. CH8 had almost 50% of the whole methylation, of the whole methyl cytosines. So this is something that I really need to investigate. So now I'm going to show a few correlations that I have done with this data. So first of all, I'm going to show you all methyl cytosines from all contexts and all chromosomes all together, like a huge data set. And then on CPG and CHG separated. So here it's everything, all my data on methyl cytosines. So we have a really high correlation between the wild species. These three are the wild species. So we have really high correlation of the amount of cytosines and its location. So they have similar methylation patterns. And here you can see that both domesticated, they have 89% of similarity too, so very high also. And between domesticated and wild, we have around 0.7578. That is a really high correlation anyway. So CPGs will have similar results. 92% correlation wild, 88 domesticated, and 70 for between species. And CHG, the same thing. Though I feel like this is a higher correlation than the other one, although I have not tested for any significance on that. Right. So on CPG context, now let's see, compare between the accessions. So those are, each scatter plot is one chromosome. So tomatoes have 12 chromosomes. And we have here one accession of the domesticated lycopersicum and here the other accession. They are pretty similar on their methyl cytosines on CPG context. So we have 94, 97% of similarity, all significant. Although we have chromosome 4, 5, and 11 with a little less correlation, but still significant. So when we compare Elsa Craig, one of the domesticated, with the other 3 wild, we can see, sorry you cannot see that good, but the correlations are also pretty good correlations. Every correlation is really high, more than 70% of correlation. And those chromosomes that were kind of different, more different between domesticated, they are really similar in all chromosomes. So chromosome 4, 5, and 11 have not like a different correlation here. And so this is now to get to the point that we could correlate the methylation, the epigenetic variation with gene expression, or the control of the gene expression and stability of the genome. So those are plots for each of the chromosomes. So here you have the position of the chromosome on the x-axis, and here you have the percentage of methylation, in red CPG, in green CHG, and in blue you have the amount of genes. So on tomatoes usually the genes are located on the edges of the chromosome. For all of them you can see this pattern on the beginning and the end of the chromosome. And when the genes are more abundant, you have less methylation on the cytosines, both on CPG and CHG sites. So in more details, the chromosome 1 here, you can see CPG, and here you can see those with 0, they are the ends on the reference genome, so it didn't have cytosines to align here. So you have here CPG having like at least 50% of methylation and really high, around 90 or 100% in the middle of the genome. CHG has more, it looks like more variable, like it can get really low levels of methylation by the end, on the edge of the chromosomes. And it's really inversal, so it's the inversal of what we find for genes. Like the genes are much more abundant on the areas where the methylation is lower. So again, trying to compare with Arabidopsis is not easy. So I have not a conclusion yet. This work is not concluded. But we can see higher methylation levels on centromeres for the 3 of the context. And we see high methylation levels in the middle of the whole chromosome, and not really areas that we could say there are centromeres. So I decided to use the data that we have on repeats that are published on Soil Genomics website. So I calculated the abundance of the repeats, and the repeats are more located in the middle of the genome and less on the areas where we have a lot of genes. So I believe that this methylation on CPG and CAG sites are really correlated to the stability of the genome, possibly of controlling or silencing transposon elements. And actually, so when you have more variation on methylation here, I could say that this methylation possibly is really correlated with the gene expression. So to finish, I look for fixed methylation states. So by that, I mean that I look at the similar states on both domesticated accessions and the same state of the three of the wild, but they would be different between them. I mean, if a position is methylated here, it should be unmethylated here, and vice versa. So I got 708 cytosine positions in that context of fixed methylated. Curiously, wild were a lot more unmethylated than domesticated, so this is one interesting thing that I'm going to look more carefully to. So only 42 were unmethylated on domestication. I wonder if wild is hypomethylated of all the genome or only on these areas. So I got 123 located near inside genes. 94 of those genes had homologs to Arabidopsis genes. 27 of these 94 were not on the gene body, located on the gene body, was before the gene, so possibly promoted regions. And 16 of this whole 94 were retrotransposons. So my perspectives continue analyzing the data in the very different ways, analyze the candidate genes with qPCRs, and possibly add more accessions to my analysis, or even the cherry tomato also, to try to understand more about the domestication of the tomato process. So thank you for everyone for this talk. So I should thank Maya at the New York Botanical Garden, Lelio, Christina, my sponsors, and Botanical Garden in Rio. It's a beautiful place. You should come and work, collaborate, or just visit. Anyway, this is our graduation program building. It's really nice. Thank you.
We have time for one or two questions.
So I didn't catch at the end when you were talking about the fixed differences between domesticated. Any evidence that those are related to fruit?
I have not seen one that said on entire database of rhabdoxin fruit development, but I got some with DNA methylation and transcription, gene transcription, so I got some interesting things to go through. So now I wanted to do the reverse, like find the genes on gene ontology and just look how it is methylated on my data set. Also, I'm going to do the other way to find interesting genes for fruit development.
Have you thought about including other Solanum species that are not domesticated to compare how methylation is associated with domestication versus just specific variation?
Yeah, this is really interesting. So I have been working with mangroves and wild species, and now that I work with tomato, it's so good to have reference genomes, transcriptomes, and then we can look more carefully on our data set. So we really want to expand this project, and maybe other wild species that could be also progenitors, but kind of has a controversy on that for tomatoes. So I have a lot of ideas to continue this.